from Microbe TV. This is Immune, episode number 77, recorded on February 20, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back. It's great to be here. It's cold.
Is it really cold there? Yeah, I bet.
It's been so warm lately, but today is spectacularly beautiful. No clouds, sun, which is unusual, but cold.
It's 3C here in New York and sunny. Yesterday I was talking to Angela Mingarelli in Montreal, where it was minus 20 Fahrenheit.
Ew. Really
cold. Also joining us from Cleveland, Ohio, Steph Langle.
Hi there. Great to be together again. I'm in a cold city, relatively. So similarly cold, but it's funny. I have colleagues on my floor who are Canadian, and they giggle at us, thinking that this is cold.
And from Madison, New Jersey, Brianne Barker.
Hi. I guess it's cold here, but in relative terms, it could be worse. 38. I can handle it.
It's been a mild winter for us. I don't know for everyone, but
here's how cold it was this morning.
Okay.
I wait for a train. I forgot. I have this little hat, right? That I put over. I forgot it.
Oh.
So I had to go somewhere where I was going to be walking for a bunch of blocks, and Macy's was on the way. I went in and bought a new hat.
Oh, wow. So it was, you were cold.
It was cold.
That's cold.
And I bought a red knit cap by Tony Hilfiger. It looks so cool.
Oh.
Red. Bright red.
We can always find you.
Yes. Yeah.
Well, that's the thing. When I come off the subway, sometimes there are students on the subway.
Yeah.
And they come up, Professor. And I say, how did you recognize me? Your hat. Everybody else has a pink or a red hat.
Good.
Okay. If you like our programs like this one from Microbe TV, we'd love your support. You can go to microbe.tv/contribute, and your donations in the U.S. anyway are federal U.S. tax deductible, because Microbe TV is a 501c3 nonprofit. We do these programs for free. We don't have ads, no monetization, because I don't like having ads, and so you get a clean experience, and we need your support to do them. So please go over to microbe.tv/contribute. Today is Cindy's turn to do a paper.
Woohoo.
So Cindy, go for it.
All right. So I picked a mouthful of a paper. It's called Trans Endothelial Neutrophil Migration Activates Bactericidal Function via Piezo-1 Mechanosensing. I thought this was really cool because if you take nothing else away, the idea is neutrophils need to come out of the blood and into the tissues in order to be able to find and kill bacteria. And this paper suggests that when they go through the endothelium, they get squished. And when they get squished, it activates their antibacterial program.
Right.
I know. I thought this was so cool. I've
Squishy toy.
taught the process of transendothelial migration a million times.
Yep.
You always kind of see in the images the cell has to get squished. But the squishing matters.
It does. It matters
the squishing
so much.
does something. That's so cool.
And it's a lot of squishing. I wore my neutrophil necklace.
Oh,
so
beautiful.
For those of you can see. So neutrophils have a multilobular nucleus. And I never thought about why that might be. So nuclei are usually nice and juicy and round. And granulocytes, like neutrophils, have these lobular nuclei that have little bubbles and then thin area, little bubble and thin area. And if you think about it, that's a pretty good idea if you're going to have to squish your nucleus.
Absolutely.
Because it's a whole lot thinner in order to get through that little spot.
Right.
That makes so much sense. I hadn't thought about that until you just said it.
Same.
And that's amazing.
Because I know somebody who studies this kind of thing, and they look at cancer cells. So cancer cells are another cell that migrates around and's got to go through really small spots, junctions. And when they do that, they actually break their nucleus and put it back together.
Wow.
And they can show this because they have labeled cells that when their nuclei burst, they can see a fluorescent version of the protein in the nucleus leak out, and then it goes back in and they reform their nucleus.
That's wild.
But yeah, so I was like, oh, they're not having the neutrophils. And I was like, oh, well then, but they have like actually smaller, thin, beads on a string nuclei that will better manage going through those tiny little pores.
Gosh, I never thought of that. That's
I am so excited about my immunology lecture for next year and getting to bring this in.
Oh,
I'm so excited.
That's cool.
Oh yeah, and that I've also mentioned this before that the vast majority of neutrophils will go between endothelial cells. So you think of a blood vessel. A blood vessel is lined in 3 dimensions with endothelial cells that touch each other, and they have adherence junctions that stick them together so the blood stays in the blood vessel and not out in the tissue, right? And the vast majority of neutrophils will find a junction between 2 cells and they'll go between that junction. But in fact, I read this and there's some cool things where the neutrophils can actually dive through the middle of a cell and come out the other side too. So that's kind of...
Oh my gosh.
What? Yeah. So neutrophils are pretty cool.
Whoa! And not that that's the point of this paper, but the diving through cells, is that endosome mediated or?
I don't know.
Okay, we don't know. That's another
they can,
episode.
I don't know that much. I should have looked into that more.
Okay.
But yeah, it's pretty cool. But most of them go around. They will roll. So neutrophils have a process that I'm sure, Brianne, as you said, you've taught a number of times. Neutrophils are flying through the blood. The blood goes really fast. And things happen, I won't go into great detail about when there's an inflammatory response, it changes the endothelial cells in the area, in the vicinity of whatever is going on, in the infection or damage or whatever. And so the neutrophils fall out of that stream, that like the fire hose that's going down the bloodstream.
It's called that shear stress, the shearing, that's that rate of flow.
So they feel a lot of stress as they're doing this, and they fall out, so it's called margination, and then they touch the endothelium and they'll slow way down and they'll kind of roll along. And they're rolling because they have attachments that attach them to the endothelial cells and they're trying to stop, but the blood flow is so fast. It has a lot of shear action on the neutrophils as they're rolling along. And eventually there's some changes that happen with some of the attachment molecules and they arrest. That's called firm adhesion. And then they search around for a junction between 2 endothelial cells and they find that and then they crawl through it and out the other side.
So cool.
And that's how neutrophils get out of the blood into the tissue. But it's this idea that these neutrophils crawl through these junctions, and that process of transmigration, because it's transendothelial migration from the bloodstream through the endothelial cells into the tissue, that actually primes them because they're going through that process. It activates their antibacterial function.
Yeah. So if anyone is super excited about this, there's a great video called The Inner Life of a Cell that goes through all the cell biology of this. And it's done by amazing animation, and I use different parts of it. There are also some really nice videos on YouTube that show this both in cartoon form and then show it with microscopy images where you can watch the cells. But I always teach the sort of, and then transendothelial migration happens after all of those other steps that Cindy mentioned, because I feel like that's something that I've thought and know much less about. And so I was excited to learn about it. And then learning that it actually, like, the squeezing matters.
The squeezing matters. That's a good title.
There you go. Okay, yeah. I was trying to think of a title. Yeah, that's a great idea.
And neutrophils are very prominent. They're one of the most, they're the first responders. There's many of them. And
So I looked up this.
Yes, go for it.
I looked this up because this is a fun fact. So each day, 1 billion new neutrophils are made in the bone marrow.
Wow. They are up to...
Each person.
Every person. Every day.
1 billion.
1 billion.
Wow.
I feel so productive.
I know.
No wonder we have to eat.
Wait, hold on. Per kilogram of body weight.
1 billion per kilogram. Wow.
1 billion per kilogram of body weight.
Wow.
And this can increase to 10 billion per kilogram of body weight during an infection, when you need to produce a lot more neutrophils.
Wow.
So I thought,
Cindy,
that's
you are
pretty cool.
improving some of my lectures so much right now.
Look, yeah.
So if you...
I looked up the fun facts.
if you have neutropenia,
It's bad.
Does the production go down? I presume so. Right?
So that could be a couple of different things. So obviously we just said the neutrophils need to get from the blood into the tissue. So a massive recruitment of neutrophils out of the blood to a site of infection, like let's say a infected lung or whatever, that can lead to a transient neutropenia.
I see.
But there's a lot of things that can lead to neutropenia. There's also we could just talk about the cool things about neutrophils all day, but they go through a maturation process. And so your body is constantly 1 billion per kilogram body weight per day. It's making them all the time. But if you have a massive need for them and it continues because you have a chronic inflammation, you can actually basically take your neutrophils that are in elementary school and release them early because they haven't completed their education.
Right.
Are there cancers where neutrophils are made that are not mature? Like leukemias,
I don't know.
Probably.
But I would guess yes.
Yeah.
The thing with neutrophils is they're very short-lived.
Right.
So they need to turn over quickly. And I usually teach this, although I should have looked into whether, what people really say about this. But I usually say it's because they are just packed full of very dangerous things. They're carrying an armamentarium or whatever you call them. Okay, help me with the word, Brianna.
Armamentarium.
Armamentarium.
I was like, I don't know what you're trying to say. Got it. Okay.
With all of this antibacterial molecules. And if they start to die, they can release those things. So they need to be made and cleared continuously.
Sure.
So that's why we turn them over.
So there is chronic neutrophilic leukemia, CNL.
Aha.
That's
Neutrophilic. They make
nu-
extra ones and
they're not
they stay
quite,
around.
they're
Yeah.
not proper. Yeah.
Yeah.
This side note, I think one of the types of immunologists we really haven't on the program is cancer immunology. Maybe we had one person in the past, but we should have more cancer-related shows. So maybe we'll look into somebody who studies that. They could come.
We should.
But those who work with neutrophils, so we have an investigator here, Christina Farr, is looking at vaginal neutrophils, and they die. You take them out of their environment and they die very quickly. You cannot freeze them like you can with other white blood cells and then bring them out of liquid nitrogen and play with them. You have to use them upon collection, and it's very challenging to work with them.
I wonder how much of it is also related to metabolic processes. They're in order to kill many microbes, they're doing things like metabolic processes that are helping to make antibacterial things, but those also might be sort of metabolic processes that do not give the cell what it needs to live for a very long time.
Yeah, sure. Definite possibility.
Yeah.
Wow. So neutrophils are very important to sustaining life and preventing infection.
Yep.
Or clearing it, rather.
Yep. And so they deploy all of these antimicrobial molecules that they have, and they die, and then macrophages clean them up. And in fact, when you have a lot of neutrophil death, it makes pus. So
that's a good point. If when you see pus, there's a lot of neutrophils in there.
Yep. Yeah.
I will also tell you, if you are ever making a lecture about all of this, and you want to put some images on your slides, do not do a Google image search for pus.
If you're easily squeamish, do not do it.
Yeah. And there can be different colors and it's different molecules that neutrophils make and everything. But the most important, so they have many, many different mechanisms. So one of them is the phagocytosis. So they will eat bacteria and they will kill them inside their phagosome. They can also what we call degranulate. So they can spit out granules that have bioactive components in it that will kill bacteria. They can also do something called neutrophil extracellular traps or nets, where they kind of spit out their DNA like a spider web and encapsulate microbes and kill them that way. So it's basically they can eat things, they can spit out granules, and they can spit out their DNA. So there's three main ways that they can kill bacteria.
I just Googled, because I knew we did an episode on neutrophil extracellular traps, and it's immune 47 if you're interested. But it's funny that because the YouTube pops up as a Google search, and when I scroll over it, it animates a little flavor of the YouTube video, and it's Cindy going.
That's funny.
Because the thing in the Spider-Man net. Yes. But I saw last year I was at a conference and I thought that the net part of it was neutrophils basically, I mean, you think about it, you spit out your DNA, that can't be a really good idea. And they're probably not going to survive, right?
Right.
But it turns out that cells can spit out little bits of their DNA, kind of like pooping them along the way. And that can be antimicrobial too. So they don't have to die when they spit out DNA, which I also thought was really cool. But yeah. Anyway, so that's interesting.
So cool.
But all of these mechanisms are cool for the neutrophils to kill bacteria, but the single most important one is production of reactive oxygen species or superoxide. And we know that this is really, really important because mice or humans with mutations in any number of, I think there's 47 different proteins that are involved in nitric oxide or reactive oxygen production, can lead to a disease called chronic granulomatous disease. And the idea is basically you can't kill the bacteria, so the body keeps walling them off into these little granulomas.
Right.
And you end up with granulomas everywhere, and eventually that compromises function of tissue, and it's really bad.
Oh, yeah.
So reactive oxygen species are very, very important.
I think those are related to the metabolism stuff.
Oh, yeah, definitely. Definitely. And the way that I looked this up too, because I always say reactive oxygen species, oxidative kills bacteria. But exactly how does that happen? I actually looked that up because I was like, I don't really know.
That shows up in micro.
Okay. So it can oxidize DNA, which causes double-strand breaks. And so that's a way it can damage DNA. It can oxidize proteins, which will affect cysteines and methionines. And that can cause unfolding of proteins and their leading structural changes and loss of protein function. And it can also peroxidate lipids and disrupt bacterial membranes. So those are like the three main building blocks of life, and all three can be affected by reactive oxygen species. Now, you might be asking yourself, well, that's a chemical. And a chemical is going to act on a lipid, a DNA, or a protein. It's not necessarily specific for bacteria, right? So we think in general that this is one of the reasons why neutrophils are short-lived, because as they deploy these mechanisms to kill bacteria, they're going to also cause damage to themselves. And damage to your DNA and things is not a really great idea, because if you survive that could lead to cancer and other things. So they're pretty rapidly cleared as they are deploying these mechanisms. If they don't die from it, they will undergo apoptosis and then be cleared by macrophages. So that's a pretty important part of the whole process.
Right.
So the next big component of this paper is mechanosensing. So I think I get excited about this. We've actually published a paper in mechanoregulation of TLR signaling.
Cool.
But I think that the physical aspects of controlling immune responses are really important and overlooked a lot. In that we usually take immune cells out of the body and we treat them with various different things, and we look at their functions, or we look at their functions in vivo, but don't really think about the environment that they're in. And there's a bunch of different physical things. Heat. In fact, heat can regulate immune cells. They work better when they're warmer. So if you think about if you get a fever, they work better.
Oh, interesting. To
a point, right? So there's you get too high a fever and it's not so good anymore. Light is another physical property. I don't really usually think about light regulating immune responses, but flow, pressure, shear stress, all of these are physical properties that can contribute to regulating immune cells. So that's pretty cool.
That's cool.
So how do you actually measure mechano things, right? So there's a lot of really cool tools that engineers have developed to look at this. So
Because the force on a cell, like the amount of force that a cell cares about is way less force than we as a whole organism care about. So how do you measure, I don't know, that many little nanonewtons or whatever?
So one thing you can do is you can find out how stiff a tissue is or how stiff a cell is by basically poking it with a cantilever that can measure the force. So this is called atomic force microscopy. That's one way to do it. You can make these devices that have little teeny tiny pillars. It's like a lawn of little, I guess like a bed of nails, and each one has a sensor attached to it. So if any one of those little sensors move in the littlest way, it can be measured. And so you can put cells down on these little micropillar fields, and you can look at how they pull and push and move and like grab onto their surroundings. So you can see how stiff tissues are. You can see how cells, move within that. You can grow cells in 2D or 3D with different gels where you have defined stiffnesses. You can control that microenvironment. And then what they do in this paper is they're thinking more about, as we said in the beginning, cells squeezing through little holes. So to do that, they can create these little chambers that have a whole bunch of different little holes in them. And so the cells get flow through one side, and the flow force is flowing them across, and then they get trapped in one of those little holes, and then they squeeze through the little hole and come out the other side, and they can go really fast. And so they can use these kind of devices. You can put sensors on either side of those channels and see how much force they have going through there. But then you can also have them where they've gone through larger pores where they don't really feel any of that squeezing force, and they can just fly right through, versus really tiny pores where they have to work really hard to squeeze through those pores, and when they come out the other side. And they can measure the function of those cells if they have gone through those larger pores or smaller pores, or not have gone through the pores at all.
Right.
So that's one approach that they use, but that's in vitro, right? And you'd really like to know, what is the difference between a neutrophil that has gone through an endothelial barrier inside the body and one that hasn't? And that's a big challenge to do that. And so they call these transmigrated cells and non-transmigrated cells. And the way they analyze this in vivo, which is more challenging, is to use some cool mice that they call ketchup mice. Like the stuff that you put on a hot dog, ketchup. And the idea is that these have knocked into a locus to the tomato. So these are cool fluorophores, right? It's a tomato. It's red fluorophore. So the neutrophils will be red. And that's fine. And you'd have red neutrophils in the blood. And if they come out into the tissue, you have red neutrophils in the tissue. But if you, for example, they're using an infection model in the lung, and if you then take out the lung, you can't really see the difference between the cells that are in the blood vessels and the cells that are in the tissues. They're all red, right? So how can they see the difference between ones that have gone through the barrier and ones that haven't? Well, they use a really clever thing where right before they euthanize the mice to collect the cells, they inject them with a fluorescent, different color, green, labeled antibody against the neutrophil surface protein, Ly6G. So what happens is there's very rapid labeling of all the neutrophils in the blood, and the neutrophils in the tissue are not labeled. So now if you take the lung out, you grind it up, and you look by flow cytometry, the cells that were in the blood are red and green because the intravenous injection of the Ly6G antibody labels the cells in the blood. And then the neutrophils that have exited the blood and gone into the tissue are only red. And they did some, experiments to show you that. And they're not the first people to kind of do this dual labeling, but they use it in conjunction with these ketchup mice. And so they can see the difference between those that have migrated and those that haven't.
Okay, so transmigrating neutrophils are only red. Non-transmigrating are green and red.
Yep.
Got it.
And so the key finding of the paper that really, they went into a whole bunch of mechanisms and everything else, but the key finding of the paper that you take away is that they collected these neutrophils that were red, so they were in the tissue, or green, they were in the lung, and then they incubated them with bacteria, and they used Pseudomonas aeruginosa, which is an important pathogen of the human lung, and they took up the bacteria equally, but the ones that had gone through the squishing, so they had gone through transendothelial migration, killed the bacteria better. So that's the key finding of the paper.
Right.
Did we actually say who the authors were and where they were from?
We did not. Do you want to give it a chance? I'll try. So the first author is Amitabha Mukhopadhyay. I think I said that right. And the last author is Asra Malik. And they are Chicago, right?
Yeah, University of
this up.
Illinois,
University of
Chicago.
Chicago. Yeah.
these authors that we mentioned did such cool work that we had to get right into talking about how exciting it was.
Yeah,
and we've missed
exactly. And the first author really is a true first author, as far as I can tell. They don't have a dual first author,
Right.
So, yeah.
And they build the story as to what exactly the function that's causing the enhanced bactericidal killing, because they do say throughout it's not an increase in phagocytosis. The same amount of bacteria are phagocytosed, and maybe not till towards the end do they say it's ROS production.
Yeah. So they went through a bunch of different things to try and get at mechanism. So first of all, they did this a whole bunch of different ways. They didn't just take cells that were labeled or unlabeled from a mouse lung, but they also used their flow device. And they showed that if you had big pores and the cells went right through, they behaved as if they were in the blood and they did not have higher antibacterial killing. Whereas if they went through small pores, they had activated bacterial killing.
Right.
And then the next thing they did was try to look at, and they did one other thing that I thought was a weird experiment, I thought. So they took these neutrophils that they had put through this device, and they went through big pores or little pores. So we think they have more or less bacterial killing. Then they put them intratracheally into a mouse, so into the throat, basically into the lung. And then they added the GFP-labeled Pseudomonas aeruginosa. And what they showed was the ones that had gone through the small pores killed more of those bacteria in vivo. I thought that was an odd experiment, but it got at the answer, sort of.
Right.
It's not really much different than in vitro, but
I'm sure it's
sitting in a mouse.
Yeah, I think it's that in vivo relevancy question. Because it would be difficult to do that, to distinguish pore sizes in the mouse. Although they did do the genetic mouse model where they were able to make the endothelial cell junctions either small or big.
Right.
So, they did address it there, but maybe just another way to prove,
Yeah. So the experiment that you just mentioned, Steph, I think is also an important one, because these cells were inside or outside, but is it really going through the junctions that's making the difference? And basically they made mice that either had a tighter junction or a looser junction. And it turns out that the neutrophils that went through the tighter junction killed even better than the ones that normally pass through. And the ones that went through the looser junctions were poorer at activating bacteria.
So, because interesting, when you say better, it's like, what are we talking about? How do we know that's relevant in any way? Right.
I guess that's what the Pseudomonas experiment was proving.
Yeah.
That
was trying to get
at in vivo killing, not just taking out cells and putting them in a dish and saying that they kill more bacteria.
Yeah. And so at this point, it's just, is there a reduction in bacterial numbers?
Correct.
Though, again, as you mentioned, we don't know why there's a reduction in bacterial numbers. We just know how good are these cells at reducing bacterial numbers.
Did you guys find that odd? Because often in figures like 1, 2, and 3, I'm like, do the ROS experiment here. It just seemed like you could take the transmigrated PMNs and just do an ROS production assay, and then in those figures you'd have your answer. It's kind of like we had to wait. I don't know if that was.
I agree.
I wasn't sure why they waited.
I agree, but also I think if you had asked me about any of this before this paper, I would have told you that the physical force didn't matter to the neutrophils. And so just the fact that they're better or worse would have been shocking to me.
True.
And so knowing like, oh my gosh, just squeezing a neutrophil makes it better is in and of itself interesting. And then we can say, well, why? And then how does it make it better? But just like, yes, actually like squeezing the neutrophil is changing its ability to do bacterial killing. Is already like, what?
Yeah. But I'm going to throw a monkey wrench in this. And then we'll talk about mechanism. And the monkey wrench is, we started by saying how these neutrophils do this thing and get out of the blood, right? So one of the very first things that happens is that when inflammatory cytokines are produced, the endothelial cells respond. And I said they change, right? And so some of the ways they change is they put up little flags, little molecules to capture the neutrophils, to have them do that, rolling and firm adhesion.
So we're talking about integrins
do,
here for aficionados.
Yes, for aficionados, it's integrin mediated. But the other thing that happens is the endothelial cells get leaky. Mm-hmm. And they get leaky because one of the goals of that whole process is to get some of those mediators that are in the blood, which include complement, into the tissues.
Right.
So one of the first things that happens is the endothelial cells get leaky, which means lighter or less tight junctions. And we just said that neutrophils are better when they go through tighter junctions and poorer when they go through looser junctions. So the whole initial thing is you try to loosen the junctions and allow the cells to get through, but we're saying that they don't kill as well when they go through those looser junctions. So conceptually, it still has a little bit of a distaste in my mouth. Like, how does this all work? But maybe it has to do with the vasculature has different areas where some are going to be leakier and some are going to be less leaky. And maybe the neutrophils that go through the tighter junction areas are activated, whereas the other ones are not. And maybe that has something to do with something. I don't know. That's hand waving.
The other thing I was thinking about, I like that. That's interesting. The other thing I was thinking about is if you are thinking about evolution and if this process would need to be changed based on the ability of a mouse to not protect itself, if there's so many more neutrophils, even though they have decreased function, but the overall protection of the mouse is the same, then it doesn't matter if they have decreased killing ability per cell because there are so many cells there, and you're recruiting other things like complement, and probably without the cells becoming leaky. Why do I have it? Look at me waving a spoon. I don't even.
I'm like wondering, are you eating?
I picked it up as if it was my pen. It's so funny. Okay, so with complement, maybe if you then knocked out complement, then you would see this big difference. So my guess is that for the organism, it did not it was not a detriment to its fitness because of other things like complement. And you're just we're making a ton of these suckers a day, so it could just keep replacing them.
Well, and that actually starts to be a reason why that experiment where you treat the neutrophils and then put them back into the mouse becomes kind of important, because you've put them into the mouse, but the mouse doesn't have the leaky blood vessels, doesn't have the complement, doesn't have all of the other stuff that, as Steph points out, is probably actually helpful. And so in that experiment, you're actually looking as, is it just the neutrophils if you can separate from all of the other
from the
stuff?
leaky? I see.
Whereas other in vivo experiments, you wouldn't be able to
that.
separate those things.
Could you also make a mouse that had a gene deletion where the neutrophils can't go through? Somehow you figure out one gene involved, right?
Oh, yeah. So there's a disease called leukocyte adhesion deficiency. And that has, it's an integrin mutation.
There's two.
There's two.
One of them is an integrin mutation. One of them is a selectin mutation.
Got it. So one is the selectin would affect its ability to attach in the beginning, and the integrin mutation, it's for the aficionados. The neutrophils, once they attach, their integrins change from low affinity, which is catch and release, and they're rolling along, to they change into a high affinity form that arrests them, and that's responsible for firm adhesion. So if they lack this ability to change from the low affinity to the high affinity, the neutrophils just roll along and then go away, and they never get out into the tissue.
Right.
And that causes a severe problem.
Right.
Because they can't get the neutrophils in to help clear the bacteria.
Interesting.
So it's a pretty significant immunodeficiency disorder.
And the one with the selectins, you also don't have a blood type because it gets rid of, it's actually
Sialyl X.
a problem with adding carbohydrates to things. And apparently it's a big problem in cows.
Oh, really?
I did not know that.
Wow.
You now know all of my facts.
You gave away all your cool facts. Now I need to watch your shit.
Yeah.
All right. So Okay, so far we have neutrophils that go through these little pores or go through endothelium, increase their antibacterial properties. But there's two main questions that this paper also addresses. One is, how do they sense getting squished to begin with? And is that important? And the second is, what are the mechanisms that squishing upregulates that allows those neutrophils that got squished to kill bacteria better? So the first part of this is a channel that's called piezo-1, Piezo-1. And if I'm pronouncing it incorrectly and somebody cringes every time, please accept my apology because I have not worked with this protein. I haven't heard somebody pronounce it before. But piezo-1 is a non-selective calcium channel. And they predicted that it was already known to be involved in tension activation of cells in general. And they figured, well, this is probably a good one. And they did a whole bunch of cool things that we don't have time to go into all the details, but they have reporter mice that basically their neutrophils will flux a color when they are having a calcium flux happening. So these are salsa mice. So they also have this fluorescent reporter, and they can do in vivo imaging of neutrophils going through the endothelial barrier, which if you have a chance to go see the one video, it's really cool. You see all these neutrophils flying along and then you see one stop, and then you see it go through the endothelial barrier. And as it goes through, it flashes because it's fluxing calcium. And this piezo-1 is this calcium channel. So they're like, that's probably what this is. And they do a whole bunch of things to show that. And there was a lot of complex modeling and engineering things. If you like that mathematical stuff, go check it out. But they figured how much tension the neutrophil was under when it was going through the pore. And they said that that's consistent with being able to activate this piezo-1 channel. And they looked at activators of this piezo-1 channel. There's something called Yoda. I thought that was kind of funny too. Yeah. So very wise old drug activates the piezo-1 channel. And so that activates the piezo-1 channel. And they could show that alone could upregulate antibacterial activity, and that neutrophils that lacked piezo-1 couldn't upregulate their activity when they went through the pores or went through the endothelium. And then they also showed that if they had a piezo-1 knockout only in neutrophils, that also affected their ability to clear the P. aeruginosa. So that whole section basically says that piezo-1 is very likely, although they didn't 100% close that loop, activated by the squeezing. That squeezing causes a calcium flux, and somehow magically that calcium flux leads to better bacterial killing. So, yep, go ahead. Did you have a comment?
I have a question, but I can wait.
Oh, yeah. No.
So one of the things that kind of left me, I had a couple questions at the end about the population of neutrophils before they transmigrate. And whether those that migrate are primed to do so, and is that subpopulation already have an increase in piezo-1 expression? Because in the beginning, I was thinking, well, how do we know that it's the mechanical force that's doing it, or are there subpopulations of neutrophils that have an ability to do this at baseline? And I don't know if we got there.
Definitely not got there. And I had not thought of that, and it is an excellent, excellent question.
Yeah, I think the thing that was sort of similar to that I had thought about was how much do we know that it's the mechanical force versus, say, a signal from the integrin or a signal from the chemokine.
Right.
That's leading to this. And I think that's one place where the pore experiment, because movement there is mediated not by integrins and chemokines, but by shear force. And so you don't necessarily have the signaling. I think that some of those help, but they don't totally address what Steph is saying of were there different populations of neutrophils? Was it a heterogeneous population that went through the pores, and only some of them had this change?
Right. I don't have because you're right. I like the experiment where it's not integrin dependent. They don't use a chemokine. So you can purport that that's the squeezing, but it still doesn't answer whether that population was predestined to do that function. And so I think ways they could have addressed that is to do RNA-seq on the population of PMNs before any migration to see, I don't know, how different are neutrophils, and are those differences related to piezo-1 expression? Because the thing about even that experiment, there's a population that moves and a population that doesn't move. How are they moving then? And what determines that ability? Trying to understand the subpopulations pre-migration, I think would be really interesting to dissect that out.
I would agree with that entirely, because we now know the more single cell RNA sequencing we do, the more subpopulations of cells we see.
Right.
And it's very hard to differentiate between cells that are predestined to do something
Right.
and something that's activated from an action. I would say the one way they address this a little bit is in their engineered flow device. All the cells are going to be pushed through a hole. And they're not necessarily integrin mediated or, selectin mediated. None of those signals will happen. It's just basically forcing a cell through a small hole.
I see what you're
and
saying.
having it fly out the other
So
side.
in that experiment, to my memory, every cell was pushed through that hole. There wasn't like a population
Correct.
remaining. And now that I'm remembering, that is the experiment where I was like, okay, I feel good about this. Right.
Yeah, because what you want to know is, do you end up with kind of two subsets after they go through the hole?
That would be cool. That would be cool.
Or do they all kind of look more homogenous?
Right.
That it was going through the hole that changed the cells,
Right. Or
Yeah.
because we know cells in general, they're not all going to be the same. And so if we can assume that the biology of piezo-1 and the squeezing can happen to everyone, how do those subpopulations respond differently?
Yeah.
Yeah.
Yeah, very cool.
So they did do some sequencing, although they didn't do single cell. They did take their cells that had transmigrated and their cells that had not from the lungs of mice. So to their credit, that's not an easy experiment to do.
Right.
And I thought they made a leap of faith.
With the CXCR4?
The NOX4, yeah.
Oh, NOX4,
so
I see, yep.
They did they show you differential expressed genes and all of this, which I thought they should have done a UMAT plot to show the general distance between the transmigrated and non-transmigrated with and without piezo-1, which they didn't do. They kind of analyzed them separately, and they make this conclusion, which was not in the top 5 or 10 genes on their list, but it was a gene on their list.
That's a good point.
Was NOX4.
Yeah.
So what is NOX? So it's a reactive oxygen species generating protein. It's part of the complex. And there's NOX2 and NOX4. And NOX2 is the one we typically associate with production of superoxide or reactive oxygen species. The complex is called the NADPH oxidase complex because they take NADP and add a proton, and then that electron is added to molecular oxygen to make an oxygen radical, and that then goes on to oxidize proteins. So that's how that functions. The interesting difference between NOX2 and NOX4, we usually think about NOX2 when we think about NADPH oxidase, and we think of it as a 2-component complex of a bunch of different, I think like 27 different proteins or something. And there's a cytoplasmic complex and a membrane complex, and the cytoplasmic complex gets activated when cells are activated and signal, and then those two complexes come together, and once they come together, they produce superoxide. The difference with NOX4 is that once that protein is made, it's constitutively active and just pumps out superoxide or hydrogen peroxide in this case. So a modification of superoxide. So they hone in on this NOX4 protein, and I don't know if they got lucky or they tried a bunch of things or what, because it wouldn't have been the gene that I picked out of the list, but they did pick it out. And they said because piezo-1 was up and calcium was up, and they also saw a HIF-1, which is a hypoxic factor, and then NOX4. They connected these things together and they said, oh, well, this must be that piezo-1 activates this HIF-1 protein, and then that is responsible for upregulating NOX4. And they provide a little evidence for that, but I thought that was the weakest part of the paper. But if you're interested in HIF-1 and those kinds of proteins, you can dig into that a little bit more. But the bottom line is that there's upregulation of NOX4. And they show that NOX4 is up when they migrate through the pores. They're up, and they originally isolated it from the lung migrated neutrophils, and they could activate with the iota to activate piezo-1 and upregulate NOX. And so they they knocked out piezo-1, and they knocked out the ability to upregulate NOX. So they pretty much showed that, in fact, when the cells migrate through these small pores, something happens inside the cell that eventually leads to upregulation of NOX4. And they could use a NOX4 inhibitor. The catch is that it's a both NOX2 and NOX4 inhibitor, and it had the expected effect on blocking the ability to kill the bacteria, but that isn't very convincing. But in the end, they, I think, have generally enough data to say that NOX4 is important, because they did use a neutrophil-specific NOX4 knockout, and they infected with Pseudomonas aeruginosa, and they showed that there was more bacterial colonies if you lacked NOX4 in the neutrophils. So it's kind of a bunch of circumstantial things to connect together that they suggest that when a neutrophil transmigrates, it activates piezo-1. Piezo-1 fluxes calcium. The calcium flux leads to stabilization of this HIF-1 factor. That leads to upregulation of NOX4. So there's production of superoxide. So then when that neutrophil encounters a bacteria and internalizes it by phagocytosis, that production of superoxide or hydrogen peroxide through NOX4 is better able to kill the bacteria. So that's the connecting story for the whole project. But there was a whole bunch of other genes that were up and down and regulated by piezo-1 and mechanosensing and all the rest of this that they just kind of ignored, and they just narrowed in on this one gene.
Yeah, so it definitely, I think you're right. I think that NOX4, the pathway that leads to NOX4 upregulation is involved. I think we could say we don't think it's the only thing that piezo-1 is upregulating to lead to greater ROS production.
Or even maybe something else. I mean, who knows what else could be changing in these neutrophils?
Sure, we didn't look at nets, right? Yeah. I mean, that wasn't...
They did look at some nets,
they did?
And they showed that in the absence of piezo-1, they had a defect in producing nets.
And so that, I guess, they went down the ROS pathway, but could they have furthered the nets to look at?
So there's a connection between ROS and nets. There's a suggestion that that in order to form nets, you require production of reactive oxygen species. So it could be connected, because if you don't have good production of reactive oxygen species, then you don't get good net formation. But I guess they didn't really convince me that there was a difference in the reactive oxygen, because I can't remember now. I'm blanking while we're talking. I don't think they ever really truly analyzed that in great detail of whether there was or wasn't a difference, because if there's still NOX2.
Right.
And NOX2 was unaffected.
Well, they do show in figure 6J in the piezo-1 knockout and NOX4 knockout, they are decreased.
Yeah.
Now, I'm not familiar with this unit, AU.
So it's an arbitrary unit. It's just a measure of fluorescence
Got
to
it.
compare.
That's why I'm not familiar.
But there were a lot of Pascals and Newtons and all the rest of that in here that we're not that familiar with. But the catch to that is that is in HL60 cells. So HL60 cells is a cell line. So it's a cancerous cell that if you treat it with certain drugs, it will differentiate into a neutrophil-like cell. And then they put that through the pore and they showed that there was less reactive oxygen in the cells if they lacked piezo 1 or NOx4. But they still had some, and because they didn't really show you compared to the non-migrated ones.
Right.
So we really can't compare. So maybe there's less, but maybe that's still perfectly a lot compared to a wild type cell that goes
I think I would have
when it goes through a pore.
liked to see in each of their experiments that have a different, using this mouse or this in vitro work. I don't know. Maybe it's not that easy to measure ROS in these systems, and that's why they were unable to do it or show it. But I would love to know, right? What if it is ROS at the end of the story? That's what they've told us. What is it in the mouse and in the ketchup mouse and these in vitro cultures? I'd love to know that, but.
Yeah, I think the thing that I was sort of interested in is we know that there is a difference in NOx4 after the squeezing, but I kind of want to know what else is different. And I could imagine a lot of things could be different. And one of them that's sort of simple is is there a difference in neutrophil longevity? We talked about how short-lived the neutrophils are. And the reason why I think about that is part of me also wonders, well, why don't the neutrophils use NOx4 before they went through why did we need to make this an inducible factor? What would be wrong with having it on all the time? And maybe the thing that would be wrong with having it on all the time is you're doing too much reactive oxygen species generation and you're shortening the lifespan of the cell.
Right.
And so you kind of have the cell not doing this. You keep the cell with a quote unquote longer lifespan. Right. But now when it has gone through a pore, it really needs to be the cavalry. It really needs to be able to work really well. And so it has to induce these things like NOx4 that may make it have a shorter lifespan. Right. And so I kind of wanted to know what other changes are happening. Maybe they're related to the NOx4, maybe they're not. Yeah. But why does it make sense to make this an inducible feature of neutrophils?
Well, I think the longevity question, but also you damage a lot of blood vessels that way.
True.
So I would have one slight counter argument to that, and that is because I think about TLRs and restricting response to when you're detecting a microbe. And it's a problem I have with this paper. And that is that if you just squeeze through a blood vessel and you start making reactive oxygen species, it doesn't necessarily mean that you're in the right place at the right time and that there's a bug there to kill.
That's true.
So if it's simply that when you squeeze through, you start making, I have a little problem with that because we think, so the last part of this whole process that we left out and didn't talk about is chemotaxis, right? So the location where the blood vessel in it is not always really close to where the infection is. And the cells need to migrate through the tissue to find that location. And they usually do that through chemotactic factors that are produced by the bacteria or cells, usually macrophages, that have encountered the bacteria and they're calling the neutrophils to that location. But if the neutrophils are now entering the tissue and they start making reactive oxygen species right away, they can damage the tissue and other cells on the way to the site of the infection. And that doesn't seem like a great idea to me. They did find another protein that was metalloproteinase, and they kind of waved their hands as to why that might be important. I actually think that is important because that will help the neutrophil get through the tissue because it needs to degrade the matrix in order to chew its way through the tissue. And it also will make a hole so that T cells and other cells that come behind it can get through that location more easily. And I think that is probably pretty important too. But they just talked about this reactive oxygen species thing.
Yeah, I will admit that one of the other things that I I was sort of curious about and was like, okay, I got a next study. I'm ready for my next study. Is the fact that, when I teach about this process of transendothelial migration, it's not just neutrophils that do it.
Right.
We can think of times where T cells undergo these same processes, for example, and they don't have the nice segmented nucleus.
Nope.
And T cells, often we think about doing their migration in a specific location called the HEV, the high endothelial venule in a lymph node. And maybe the idea is that the HEV isn't so squeezy.
It's very much not squeezy. We already know that it's very leaky. Mm-hmm.
But so maybe the idea is that the force isn't as much of a problem there. Mm-hmm. Or maybe T cells are getting cool signals from this force thing as well. And so I would love to see that in a next study.
I bet squeezing T cells changes their function for sure.
Now I'm going to get really little tweezers and just go around and squeeze all my cells all the time.
See if they release something.
All right.
Go ahead, Vincent. Yep. Mm-hmm.
I just said we have to wrap it up.
Yep.
Did you have a
well, the
final
only thing, my final thought, so to Cindy's comment, I think because the chemokines, though, they are selecting neutrophils to the site in blood because those chemokines are secreted into blood or they're expressing on that side of the endothelium to help. So I do agree with you. You don't want to pre-activate too early, but maybe that's taken care of because they're getting to that site because of the chemokines. I don't know.
Yep.
That's true.
Yeah.
That's true.
This is great. I always show a slide of rolling and all the receptors involved, and then the diapedesis.
Yep. That's another word for it. Yeah.
I never thought of mechanisms, and this is great because it opens my eyes that to squeezing through actually does something. I think that's terrific.
Yeah.
Thank you, Cindy.
Yeah, thanks, Cindy.
That's Immune77. You can send any questions or comments you might have to immune@microbe.tv. And if you enjoy our work, consider supporting us. Microbe.tv/contribute. Cindy Leifer is at Cornell University. Cindy Leifer on Twitter. Thank you, Cindy.
Thank you.
I don't use X. I just like Twitter. I'm sorry. Steph Langle's at Case Western Reserve University. Stephanie Langle on Twitter. Thank you, Steph.
Thank you. Thank you, Cindy. This was great.
Yeah.
And Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thank you. This was awesome. I learned so much. Cindy, thank you for picking this paper. I'm so glad we decided to geek out about neutrophils.
Awesome.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.